Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is September 23rd, 2024, and we're talking to Andy Groves. Andy is professor and Vivian L. Smith Endowed Chair in Neuroscience at the Departments of Neuroscience and Molecular and Human Genetics at Baylor College of Medicine. Andy's lab studies the development of the inner ear and of hearing, and the potential for its repair after damage. To do this, he studies the molecules that control cell development and differentiation in the inner ear and the mechanisms controlling expression of those molecules. Hi, Andy.
Hi, it's very nice to be here.
Welcome. It's great to have you. Also with us is our own Marina Silveira, who studies the auditory system. Hi, Marina.
Hi, very happy to be here.
Been on the podcast a lot lately.
A lot lately, yeah.
So mammals, Andy, including us, suffer from hearing loss just caused by the accumulation of damage caused by loud noises, medicines we take, and lots of other things, probably. And most of our hearing trouble stems from losing our auditory receptors, which we don't grow back. But in some animals, like birds, the receptor cells do get replaced when this happens. But not in us. Let's start off with a summary of what's the problem. Why can't we be more like birds and grow our
Well, that's the million-dollar question. So, as you said, in all vertebrates, we detect sound, and indeed we detect balance and vestibular information with these receptor cells called hair cells. So these are mechanoreceptive cells. So they are responding either to the vibrations produced by sound waves, or in the case of our balance system, by the force of gravity or by the motion of our head that detect both linear and angular acceleration. And these cells are incredibly sensitive. They are capable of detecting motions at the nanometer range. And the problem is they are easy to damage. And so, as you said, loud noises, the wear and tear of old age, certain kinds of drugs, aminoglycoside antibiotics, chemotherapy drugs that contain platinum, kill these hair cells very effectively. As you also mentioned, so non-mammalian vertebrates can regenerate their hair cells. And this process is a classic regeneration. So if you look at any vertebrate, either in their auditory system or in their balance system, hair cells are surrounded by accessory cells called supporting cells. And in a sort of neuroscience context, you can think of them as being akin to glial cells. And what happens in all non-mammalian vertebrates is when a hair cell dies, something, and this is the million-dollar question, makes those surrounding supporting cells promote hair cell regeneration. Typically, this is caused by a supporting cell dividing, and then one of its daughters turns back into a hair cell again. Honestly, at a population level, we're not quite sure exactly whether every cell behaves like that. But in a bird, for example, the functional outcome of that is that a bird will restore its hearing. Similarly, in a fish, if you eliminate its water-detecting hair cells, they will lose their sense of detecting water flow. After regeneration, that sense will be restored to them again. And so really the question that everyone wonders about is, what is it that is allowing those supporting cells to re-enter the cell cycle and then undergo some kind of decision such that one of them turns back into a hair cell? And it's important to understand that in many of these non-mammalian vertebrates, this replacement and turnover is happening all the time in at least some parts of the inner ear. So, for example, in birds, the balance organs that detect gravity and linear and angular acceleration, those organs are turning over at a slow but steady rate all the time, in a manner that's analogous to our skin cells or our gut cells turning over in a coordinated and regular ongoing fashion.
Maybe taste cells are an even better.
Yes, that's also correct, yeah. And so this turnover doesn't have to happen just after damage, but it's been observed that in birds, for example, the bird hearing organ does not normally turn over. But if you damage a bird hearing organ with intense sound or with some of these drugs that kill hair cells, you see a rapid recruitment of supporting cells within, a day or two to start dividing and then restore the missing hair cells. And it's been shown in at least some studies that you can do this in birds repeatedly. So you can put a bird through successive cycles of killing the hair cells regenerate again, you can kill them again, they regenerate again, and so on. I don't know exactly how many times people have tried this, but it's at least several times. And this suggests that this property of regeneration is not a transient process, that the supporting cells are capable of mobilizing again and again to restore the hearing organ back to a functional state.
So, do we also lose our vestibular hair cells and gradually
Absolutely.
have balance problems?
And indeed, different people will cite different statistics, but a sort of ballpark statistic is by the time people reach the age of about 65, sort of around retirement age, at least half the population in the US will report some kind of hearing loss. Now, given the reluctance of people to admit that they have a hearing loss, that number is probably higher.
You should ask their spouse.
Exactly. Yeah. And well, that also gets onto the topic of selective deafness, which we won't touch on. What people suggest is by the age of 75, at least half of the US population will experience balance problems, falls, and so on. Again, it's hard to exactly, quantify that and measure that. But again, not to trivialize this, but if you talk to older people, there are two big fears they have. One is dementia, and the other is falling over and breaking a hip. And indeed, there is an age-dependent loss of hair cells in the balance organs of the mammalian ear. There have been some studies in post-mortem samples of humans aged between 75 and 85, and it's actually frightening in some of those patients how few hair cells are remaining. And so, you then ask, well, how are we able to negotiate our way in the world at all? And that's because this gradual loss of vestibular input can be compensated by, for example, visual cues, by proprioceptive cues. And that's a common observation, that there can be this compensation. Now, you can uncover those defects by performing vestibular testing on people. And indeed, for example, people with balance problems often do a lot worse in the dark when they don't have the visual cues to steady them. And so I think damage and loss of vestibular hair cells is equally a problem in mammals in general and humans in particular. It's just we're better at compensating for it than when we lose our auditory hair cells and begin to lose our hearing.
So your approach to fixing this is not that different from that of, many people who talk about cell therapy in our podcast and about epigenetics. But one of the remarkable things about it is that you're doing it in vivo, at least part of the time. So instead of taking a fibroblast out, and programming it to become a hair cell, and then trying to then insert that hair cell into the inner ear, which I'm sure you could tell me why that would not be a good strategy. Instead, there are still this body of supportive cells left around in the inner ear, and the idea is to get them to convert. So what's the strategy for for doing
Well, so just briefly to touch on the question of transplantation, because I think it is something that is brought up from time to time. So obviously there are a whole variety of disorders or degenerative conditions where people talk about transplantation. And what I sometimes do is, if you visualize the spectrum of how well transplantation works, at one end of the spectrum, you have bone marrow transplantation. Obviously, highly successful. People have bone marrow transplants. And I don't want to get into trouble by oversimplifying this, but essentially, when you are implanting bone marrow into a patient, to the first approximation, so long as you're getting it into the bone, the precise location or precisely how many cells you transplant doesn't particularly matter, because you can recreate this niche for hematopoietic stem cells to proliferate and do what they would normally be doing. And so spatial accuracy and getting the right number of cells is not an immediate problem. At the other end of the spectrum, you have the cochlea, okay, an organ of extreme mechanical sensitivity where under normal hearing conditions, the membrane on which the hair cells are sitting is vibrating up and down by between 5 to 10 nanometers. And the arrangement of hair cells and supporting cells in that organ is absolutely invariant and precise. That's one of the reasons that I, as a developmental biologist, like to study DNA, because when you see these beautiful cellular patterns, it means there are very precise developmental rules that are going on to set that pattern up. What that means, though, is the idea that you would be able to just sort of inject cells into the cochlear duct and hope that they would go to the right place in the right numbers without damaging or compromising the exquisite mechanical properties of the cochlea, that's a bit of a stretch, I think. Obviously, as scientists, we never want to say never, but from the point of view of transplantation, I would say bone marrow is at the most feasible and clearly, demonstratively feasible approach to transplantation. I would say that the hearing organ, the organ of Corti, is really at the other end of the spectrum of possible. So, given that, if we can't do it from without, how do we do it from within? And that's where a variety of labs take the approach, well, what can birds and frogs and fish teach us? What is it about those supporting cells that allows them to undergo these regenerative processes, but mammals cannot? So something that a number of labs, including ours, discovered, let's say about 20 years ago now, is that, in fact, in young animals, in young mice, mouse supporting cells do have a transient capacity to both divide and make hair cells, produce new hair cells. The problem is that that capacity is rapidly lost, and in the case of mice, is lost prior to the onset of hearing. So newborn mice, unlike humans, cannot hear. They start to develop their hearing at about 2 weeks of age. And what my colleagues and I found many years ago now is that essentially that regenerative window is lost in the first postnatal week prior to the onset of hearing. So in humans, although it's sometimes difficult to make exact comparisons, but in humans that might be in the 5th month of pregnancy. So well before a human baby is born, we would guess that the regenerative potential of the hearing organ would be lost. And so
by that time you must have all the hair cells you need,
exactly,
and all your,
yes.
at least all you need to get started.
This is the fascinating thing. In a newborn mouse, you have the full complement of hair cells and supporting cells. And during that 2-week period, what is going on is not the addition or subtraction of cells, but rather both the hair cells and their surrounding supporting cells are maturing. And in fact, the morphological maturation of both the hair cells and the supporting cells, this post-mitotic differentiation, if you will, is actually a beautiful process. If you study with scanning or transmission electron microscopy, just the morphological changes in those cell types over that 2-week period are absolutely beautiful. And it is almost guaranteed that that morphological specialization leads to the exquisite mechanical sensitivity of the organ of Corti. What we have, and my colleagues have tried to do, is to ask what are the transcriptional and epigenetic changes that are happening in supporting cells during that first postnatal week or so that removes their ability to regenerate. Clearly, if you're not changing the cell population, if you're dealing with the same cells that can regenerate at birth versus the same cells that refuse to regenerate a week later, you've got to look inside those cells to see what's going on. And one of the things, but by no means the only explanation, but one of the things we find is that if you look in supporting cells and interrogate the genome and ask, are hair cell genes transcriptionally accessible? Is their epigenetic signature compatible with being able to switch a gene on? You find that at birth, many hair cells, even though they are not transcribed in supporting cells, are at least in an accessible state. And what my colleagues and I have found is that over a period of just a week, those hair cell gene loci become transcriptionally inaccessible and epigenetically silenced progressively using different mechanisms in supporting cells. But the cumulative effect of that is to, we believe, to at least contribute to the failure of supporting cells to be able to turn back into hair cells in mammals. What people are starting to do is to do similar experiments in birds and fish now. And some of the tools to do that took a little bit more work to get going in those organisms. But I think the consensus is starting to be that that window of opportunity to regenerate on the basis of epigenetic availability of these loci is prolonged in those species. And so the question, one of the therapeutic questions at least, would be how could we bring that back in mammals, and whether that would be sufficient to promote regeneration or not. And we don't have the answer to that yet.
So kind of to complement what you're saying, have people done like comparative studies looking like bird, frogs, and fish and mammals, kind of like RNA, single cell RNA-seq, and see what are the difference, what different gene expressed across species that could be driving regeneration?
That is an excellent question. And in fact, our lab is part of a consortium funded by the Hearing Health Foundation. The consortium is called the Hearing Restoration Project. And indeed, one of the studies that we are finalizing right now is actually a comparison using single-cell RNA-seq of fish hair cells, chicken hair cells from both the auditory and the balance organs, mouse hair cells from the auditory and balance organs, indeed some human hair cells from post-surgery samples, asking what is the common transcriptional signature of hair cells? Are there distinct signatures in hair cells that respond to sound versus cells that respond to, balance stimuli? Where that work is going to go, obviously, is in the epigenetic sphere. If we can do the same thing for supporting cells, can you start to uncover epigenetic differences at the supporting cell level in these different species, and indeed between different organs in one ear? Because I forgot to mention that in mammals, there is some evidence for hair cell regeneration in mammalian balance organs. It's not very good, and certainly not enough to promote functional recovery in humans or mice.
So those are two different ways. One is to look at the animals that don't close the door on regeneration versus the ones that do. And the second is to look at the ages within like a mouse.
Yes.
In which regeneration is possible, and then when the door closes. And presumably, those will give you related answers or complementary pieces of the puzzle, I guess. But you already found some identified epigenetic factors that control this, at least in the aging, in the postnatal mouse period. So, how about, can you give us a quick rundown on that without overwhelming us?
Sure, yeah. So we are not epigenetic experts, so we have just begun to scratch the surface in this. But for example, it is known that certain histone modifications are associated with, transcriptionally accessible chromatin with less permissive environments, and obviously DNA methylation is the final nail in the coffin in terms of a region of the genome being active or silenced. What we have found, and we do this work by purifying populations of supporting cells and then carrying out measurements of epigenetic state, whether that's just looking at chromatin accessibility in general, using a technique called ATAC-seq, or profiling particular histone modifications. And to sort of cut a long story short, what you see is initially what we would call a decommissioning of enhancers, so removing histone modifications that are associated with accessible and transcriptionally active chromatin, replacing those with histone modifications that tend to be associated with more quiescent or silent regions of the genome. And then in even older supporting cells, we find evidence for the establishment of domains of DNA methylation. So it's not simply one process comes in and switches off the regenerative capacity. It is a series of histone and DNA modifications that eventually lead to the tissue or the hair cell loci in those cells, the supporting cells, being placed beyond use. And this is not unique to hair cells. Obviously, in many regenerative systems or many organ systems that have an initial capacity to regenerate that is subsequently lost, you see similar processes. For example, in the heart, you can lesion a newborn mouse heart, and the cardiomyocytes are mobilized and mount a regenerative response and will repair the injury very rapidly. You perform that same lesion in a 7-day-old mouse, and once again, just like you see in the ear, the cardiomyocytes are incapable of mounting that response. And just like what we're doing in the ear, people are looking at the transcriptional and epigenetic changes that are occurring in those cardiomyocytes that prevent regeneration. The challenging thing for the ear, and this is always a trade-off when you work with, the inner ear. It's a beautiful organ. It's incredibly good at detecting, tiny vibrations or movements, but it has very few cells. So in a mouse cochlea, there are only 3,000 hair cells. And as the animal gets older, the entire inner ear becomes encased in the strongest bone in the body. And so I often have, for want of a better word, organ envy. I sometimes wish, why didn't we decide to work on the cerebellum or something else where you have the cells are much more plentiful and they're much more accessible? Obviously, we are working on a beautiful and morphologically elaborate organ that we love. It's just we don't have many cells to play with. But what that means is that it is much harder for us to do these kinds of molecular biological experiments that are always made easier by having a ready supply of cells. And technology is making life easier for us. When we first started this, 20 years ago, doing microarray experiments, postdocs in our lab would do heroic experiments where they would dissect hundreds of cochleas. And now, obviously, those experiments are much easier. I wouldn't say they're trivial. We still encounter problems with small cell numbers, and working in mature adult animals is still challenging because before you do anything, you have to crack open this bone before you can get your hands on the cochlea.
Well, I'm worried a little bit about the scope of the epigenetic problem, because from a therapeutic point of view, you have to ask, are there going to be 2,000 genes that we're gonna have to manipulate, or is it going to be 4? Sometimes I'm really impressed by the small number of things you have to add to a fibroblast to turn it into a neuron or something like that. And you think, gosh, only those, like 4 transcription factors are enough or something? But that makes its own mystery, like how 4 could possibly do it. But you hope it's going to be a small number, right? Because if you're going to try to modify something, what's the ballpark?
So this touches on another area of our work, which is trying to reprogram supporting cells into hair cells using transcription factor reprogramming. And what we find is that in young animals, a single hair cell transcription factor, the gene is called ATO1, is capable of reprogramming non-sensory cells very beautifully into morphologically quite impressive and at least partly functional hair cells. Once again, we find that in older and older animals, one factor isn't going to do the job. Right now, we are working with 3 hair cell transcription factors. We do find that the expression of those in adult supporting cells in mice that have been experimentally deafened can convert them into what I am going to call hair cell-like cells. But they are by no means functional, morphologically normal hair cells. And this raises the question that I think is a salient one for many forms of cellular reprogramming, which is in order to convert one cell type into another, you don't just want to activate the ensemble of genes of your target, in other words, the full panoply of hair cell genes. At the same time, you're going to have to silence the genes in your starting cell population. So, in other words, you don't just want to switch on hair cell genes, you need to switch off supporting cell genes. And I think people debate as to what is the best way to do that. So some people believe that you may first need to restore or revert your target cell population, in this case a supporting cell, to maybe a somewhat of a more primitive state, to then open up more of the potential to go down a different developmental lineage and turn into a hair cell rather than a supporting cell. Other people are thinking more about maybe there are microRNAs that are specific to hair cells that normally function to silence other gene regulatory networks that you don't want to be present in the hair cells. Maybe you're going to need, in addition to transcription factors, maybe you're going to need microRNA-mediated silencing of supporting cell gene networks. So there are many ways, conceptually, to turn one cell type into another. And I think the goal, the elusive goal, is to find, as you put it, the smallest number of factors that could achieve the greatest and sort of highest fidelity reprogramming of one cell type into another. Right now, we're absolutely nowhere near that. And I think one of the challenges for this field is it's very easy to use, or comparatively easy, to use in vitro systems, cell culture systems, organoid systems, to produce immature hair cells. We can do that, or the field can do that, relatively easily. The challenge is to show that those in vitro systems can develop morphologically complex, mature, functional hair cells. And I think that has been much, much harder. And so this is why, even though it's challenging, we devote a lot of our energy to this in vivo conversion and reprogramming rather than trying in vitro systems. I think there are absolutely places for those, but it's not something that we are trying in the lab right now, although excellent progress is being made by other labs in that area.
So controlling the expression of transcription factors or various other kind of epigenetic factors in the cell in vivo, though, is tricky. It's not like you can just pour them on and expect them to work. So you have to express them by genetic manipulations in the cells themselves.
Yeah.
So what's the trick? Like if you've got 4 different molecules that you would like to express in a hair cell, and specifically only in a hair cell, or specifically only in a supporting cell, how do you do it?
So you are elegantly and brutally laying out the problem that the trick for regeneration is to express these reprogramming factors in the right cells at the right time, likely for a transient period. For example, the hair cell transcription factor I referred to earlier, ATO1, is only transiently expressed in hair cells. So in the mouse, hair cells start being born at about embryonic day 14, but a week later, shortly after birth, ATO1 is extinguished from hair cells. So it is one of these transcription factors that sort of acts as the starting gun, but its presence is not continuously required in hair cells after they become functional. And indeed, it is possible that its continuous presence would actually be harmful or at least suboptimal to produce a functional hair cell. So not only do we have to target the right cells in the cochlea for reprogramming, but we have to achieve the right levels of expression, and we probably have to do this transiently. So those are big challenges. We don't pretend to try and solve all of them at once, but we are starting to experiment with a variety of different approaches. So, for example, AAV technology is, like in many systems, is successful in the inner ear. And in fact, in the last 6 months, there were some incredibly encouraging gene therapy trials using AAV to correct a rare form of genetic deafness with a gene called otoferlin. And the results of those clinical trials, carried out by different groups independently, were incredibly encouraging. So AAV clearly is a feasible method of delivering forms of gene therapy to the inner ear. We're now starting to combine that with cell type specific promoters or enhancers. So you might achieve a broad tropism of AAV infection in the cochlea, but then restrict expression to particular cells or particular times by careful selection of gene regulatory regions, enhancers or promoters. We're also now starting to experiment with modifying messenger RNAs to make them more or less stable, with the idea that you could transiently stabilize a messenger RNA using a drug or some kind of ligand to stabilize it for a brief period of time, then withdraw the drug, shut off the gene therapy, and then hopefully allow more normal hair cell regeneration to take place. Now, all of these are speculative experimental technologies, but honestly, that's where my lab is more comfortable with. We try to demonstrate that things will work in principle, and I hope that one day maybe 1 or 2 tiny things that we discover might be taken up by people who are much better at implementing these technologies in an actual therapeutic situation.
Andy, could you comment a little bit on your thoughts about evolutionary, like why do mammals don't regenerate hair cells versus, but what is the difference there? I know this is hypothetical, but
Sure.
What are your thoughts on it?
I mean, I get asked this question a lot. Why did we lose the ability to regenerate? What is the advantage there? Why wouldn't that be selected against? And the hand-waving argument that I come back with is that in the natural world, there are no rock concerts, there's no industrial machinery, there are no chemotherapy drugs, there are no aminoglycoside antibiotics. One of the few things that occurs in the natural world that will kill hair cells is old age. And from a brutal perspective, evolution does not care about old age so long as you've already reproduced. And so any negative consequence of not being able to restore your hearing with old age, honestly, would probably not be under strong selective pressure. Okay? Now, that doesn't answer the question of why mammals lost the ability. And again, we don't know for certain. People have different ideas. One possibility, if you look at the hearing organs of mammals versus non-mammals, the arrangement of cells, the number of cells, and the specialization of both hair cells and supporting cells is significantly greater in mammals compared to birds and frogs and fish. And so if you look at the mammalian organ of Corti, in any cross-section, you have exactly one inner hair cell that receives afferent innervation, sends signals to the brain. You have exactly 3 outer hair cells that participate in the amplification of sound to make the organ of Corti extremely sensitive. If you look at a cross-section of a bird hearing organ, you have lots and lots of hair cells spread across that cross-section. So clearly the precise numbers of hair cells and supporting cells has to be related, at least in part, to the extreme mechanical sensitivity of the mammalian cochlea and its ability in some animals to detect sounds up to over 150 kilohertz in sound frequencies, which, with the greatest of respect to non-mammalian vertebrates, they can't do. Birds can do amazing things with their hearing organs. So an owl can hunt and catch prey in total darkness. They are expert sound localizers, but owls can't hear above about 10 kilohertz.
Yeah.
And so one speculation is that in order to produce a hearing organ with precisely the right number of hair cells and supporting cells, during the course of evolution, in order to generate that structure, you had to really clamp down on the potential of that organ to generate new hair cells and supporting cells. Now, that's a hand-waving argument. Yeah. We can't really test that. Interestingly, one possible area we could test it, if you look at egg-laying mammals, so the dark-billed platypus and the spiny anteater, they actually have an organ of Corti that looks similar to ours. They have inner hair cells, they have outer hair cells, they have more rows of them, but the cell types are actually there. They have pretty poor frequency discrimination, so they have low frequency hearing. And so what is interesting there is that the cell types, or the basic cell types, probably arose before high frequency hearing came about. So obviously people have asked, well, why can't you, do damage to a duck-billed platypus and see if it can regenerate the hair cells? Well, 2 things. Firstly, the platypus is a protected species. Secondly, the platypus is the one mammal that is venomous. So you probably don't want to mess around with them for both legal and safety reasons. But it would be actually fascinating to test that.
Thank you very much, Andy Groves. That's been incredibly fun.
Yeah, fantastic.
And thanks for being on Neuroscientists Talk Shop. And Marina, too. Thanks for joining us.
Thank you.
My pleasure.
This has been Neuroscientists Talk Shop.